Hello everyone and welcome to our channel, Mootechie. It's so wonderful to have you here. Today, we're going to make the world of fats and lipids simple, visual, and fascinating. Get ready to see the building blocks of life in a whole new way. Now, let's begin our visual journey into the world of biochemistry. Here we are in the world of biochemistry. Today's topic is the definition, structure, and functions of fats. We'll cover the definition and structure, the classification of lipids, the difference between saturated and unsaturated fats, and their crucial biological functions. Let's look at this wonderful illustration on the slide. It's showing us the molecular world of lipids. On the left, we see the structural formula of a phospholipid, specifically phosphatidylcholine. Next to it is a space-filling model, which gives us a 3D view of the same phospholipid molecule. It's a beautiful depiction of how these molecules are put together. We can see the fatty acid chains, and a generic, simplified depiction shows a phospholipid molecule with its two distinct parts, a hydrophilic or polar head and the hydrophobic or nonpolar fatty acid tail. Another view zooms in on a single fatty acid molecule, again with that hydrophilic head and hydrophobic tail. Finally, we see how these molecules assemble in nature to form the iconic lipid bilayer, the fundamental structure of all our cell membranes. This illustration is provided by encyclopediabritannica.com. So what exactly are fats or lipids? First, their defining feature, their hydrophobic nature. Lipids are a diverse group of organic compounds that are hydrophobic. This means they repel water and do not mix with it. Think of oil floating on top of water. Now, their composition, the fats we commonly think of called triglycerides, have a specific structure. They consist of one glycerol molecule attached to three fatty acid chains. Because of their structure, they have unique solubility properties. They are insoluble in water, but dissolve readily in organic solvents like ether, chloroform, or benzene. And the lipid family is broad. The major types include true fats, phospholipids, like the ones we just saw forming bilayers, steroids, such as cholesterol, and protective waxes. Let's break down that general structure of a triglyceride. Imagine it like a little letter E. The central part is the glycerol backbone. This is a central 3-carbon alcohol molecule that serves as the structural foundation or anchor. Attached to this backbone are 3 fatty acid chains. These are long hydrocarbon chains typically containing 12 to 24 carbon atoms, and they are almost always even numbered. In unsaturated fatty acids, the chain can contain double bonds. These double bonds can be in one of two configurations: a cis configuration, which creates a bend in the chain, or a trans configuration, which keeps the chain relatively straight. This structure has built-in variability. The fatty acid chains vary in their length and in their level of saturation. It is this variation that determines whether the fat is solid or liquid at room temperature. Let's meet the components, glycerol and fatty acids, up close. First, glycerol, with the chemical formula C3H8O3. It's a simple polyol compound containing 3 hydroxyl groups, or OH groups. These groups are responsible for glycerol's solubility in water. Then we have the fatty acid chain. A fatty acid consists of a long hydrocarbon chain with a carboxylic acid group. That's a COOH group. These chains are quite long, typically containing 12 to 24 carbon atoms, and in nature, they are almost always even numbered. In unsaturated fatty acids, the chain can contain double bonds. These double bonds can be in one of two configurations: a cis configuration, which creates a bend in the chain, or a trans configuration, which keeps the chain relatively straight. Next, we explore a helpful classification overview for lipids. We can classify them in several ways. First, by saturation. This divides them into saturated fats, which have no double bonds in their chains, and unsaturated fats, which have one or more double bonds. Second, by bonds within the unsaturated category. Unsaturated fats are further split into monounsaturated, with one double bond, and polyunsaturated, with more than one. Then we have the lipid class. This includes triglycerides, our main energy storage form, phospholipids, the builders of cell membranes, steroids like cholesterol and hormones, and protective waxes. And finally, by physical state. We often categorize them simply as fats, which are solid at room temperature, and oils, which are liquid at room temperature. Now, let's visualize the key difference saturated versus unsaturated fats. On one side, we have saturated fats. Their chains have only single bonds, which makes them straight chains that can stack together nicely. This tight packing is why they are solid at room temperature. Common examples are butter, meat, and lard. On the other hand, unsaturated fats, their chains contain one or more double bonds. These double bonds, especially in the cis form, create kink chains that cannot stack tightly together. This loose structure is why they are liquid at room temperature. Think of olive oil, fish oil, and nut oils. But let's go deeper into the unsaturated category, monounsaturated versus polyunsaturated. For monounsaturated fats, or MUFAs, the classic example is oleic acid. It contains exactly one double bond. These fats are liquid at room temperature, but may turn solid when chilled. They're fantastic for supporting healthy heart function. You'll find them in olive oil, avocado, and peanuts. Now, polyunsaturated fats, or PUFAs, these contain two or more double bonds. An example here is linoleic acid. This category includes the famous omega-3 and omega-6 fatty acids. These are essential fats, meaning our bodies can't make them, and we must get them from our diet. Great sources include sunflower oil, walnuts, and fatty fish. But there's another twist in the unsaturated story: cis versus trans fatty acids. This is all about the geometry around the double bond. In a cis configuration, the hydrogen atoms are on the same side of the double bond. This causes a pronounced kink or bend in the fatty acid chain. This is the configuration of most naturally occurring unsaturated fats. Examples include natural vegetable oils. In a trans configuration, the hydrogen atoms are on opposite sides of the double bond. This results in a straighter chain that mimics the structure of a saturated fat. This configuration is mostly artificial, created through an industrial process called hydrogenation. Examples are hydrogenated fats, often found in some margarines and processed foods. Now, let's meet the major lipid types and their unique jobs. First, triglycerides. These are the primary form of fat storage in the body, composed of that glycerol and 3 fatty acids we keep talking about. Their primary function is energy storage. Then phospholipids. These are amphipathic molecules, meaning they have both a water-loving and the water-fearing part. This amazing property allows them to form the structural bilayer of all cell membranes. Next, we have steroids. This family includes cholesterol and hormones like testosterone. They are characterized by a distinctive 4-ring structure. Their key roles are in creating hormones. And finally, we have waxes. These are long-chain fatty acids linked to long-chain alcohols. They provide water-repellent coatings in nature, serving a crucial role in protection like on plant leaves or in your ears. So what do all these fats do for us? Here's an overview of the functions of fats. 1, they are a major energy source. Fats are a dense fuel reserve, providing 9 kilocalories per gram. That's more than double what carbohydrates provide. Fats are a dense fuel reserve. This highly concentrated energy is ideal for long-term survival and endurance activities. They're stored in a compact way without water. This anhydrous storage makes fat a lighter and more efficient long-term energy store than glycogen, which holds a lot of water. This storage happens in adipose tissue, specialized cells that stockpile large amounts of triglycerides for future needs. When the body needs energy, it initiates a process called lipolysis, where fat is broken down and mobilized during fasting or prolonged exercise. Moving on to the structural functions of fats, which are absolutely vital. First, the phospholipid bilayer. This forms the foundational barrier of every single cell. The amphipathic nature of phospholipids, with their hydrophilic heads and hydrophobic tails, creates a stable membrane structure in our watery bodies. Embedded within these membranes are cholesterol molecules. They modulate membrane fluidity, preventing the membrane from becoming too rigid in the cold or too permeable in the heat. And we can't forget myelin insulation. Fatty myelin sheaths wrap around nerve axons, acting like the insulation on an electrical wire. This dramatically increases the speed of electrical signal transmission in our nervous system. And we can't forget organ protection. Visceral adipose tissue acts as a natural shock absorber, cushioning vital organs like the kidneys and heart from everyday bumps and impacts. The diagram on the slide beautifully illustrates that. We see the phospholipid bilayer with the hydrophilic heads facing the extracellular fluid and the intracellular fluid, and the hydrophobic tails tucked safely in the middle, creating a stable barrier.
Fats are also master regulators. Let's explore their regulatory and transport roles. They are crucial for creating steroid hormones. Lipids like cholesterol are the precursors for steroid hormones such as estrogen and testosterone, which regulate everything from metabolism to reproduction. Then we have eicosanoids. These are signaling molecules derived from fatty acids. They act locally to control important processes like inflammation, immunity, and blood clotting. Dietary fats are also essential for absorbing fat-soluble vitamins A, D, E, and K. These vitamins regulate critical functions like vision, bone health, and protecting our cells from oxidation. And since fats don't mix with blood, how do they travel? They hitch a ride on lipoproteins. These are complexes that transport lipids like triglycerides and cholesterol through our water-based bloodstream to all the tissues that need them. We've mentioned them, now let's highlight the superstars, essential fatty acids. Our bodies can't make these, so we must get them from food. We have two main families, omega-3 and omega-6. Omega-3s are found in foods like fish, flax, and chia seeds. They come in forms like ALA from plants and EPA and DHA from marine sources. They are critical for building cell membranes and for anti-inflammatory pathways. Omega-6s are found in nuts, seeds, and oils. Key forms include linoleic acid and arachidonic acid, which is abundant in vegetable oils and processed foods. These fats play vital roles in brain development, heart health, and regulating the body's inflammatory response. However, balance is key. Modern diets often have an excess of omega-6, so improving the omega-6 to omega-3 ratio is considered very important for reducing chronic inflammation and promoting long-term health. And that brings us to the end of our lesson. Thank you for joining me. Here's a quick recap of what we covered today. We learned the definition. Fats are hydrophobic triglycerides made of glycerol and 3 fatty acids. We explored their classification, saturated versus unsaturated, and cis versus trans configurations. We discussed their vital functions, from energy storage and insulation to organ protection. And we saw their role in regulation, in hormone synthesis and vitamin absorption. This is Mootechie, making biochemistry simple and visual. If you enjoyed this journey, please subscribe for more. Thank you. See you in the next class.